Hello, my name is Curtis. This presentation is a differential gene expression associated with the pathogenesis of schizophrenia. So a little bit of introduction to schizophrenia. It's a genetic disorder. It also has environmental factors. And if there's a stressful event in the environment or you have a prolonged stressful event, it could be a trigger for schizophrenia. And certain symptoms may include hallucinations, delusions, abnormal behavior, disorganized speech. There isn't any known treatment for it. There's various antipsychotics that can be used to control the symptoms, and it's different for everyone. Some people the medication will not work, and there will be a trial and error process where the doctor tries to figure out which medication will work for that person. But there are certain comorbidities with it, such as social withdrawal, substance abuse, anxiety and depression, panic disorder, OCD, and PTSD. So what we're going to try to do is compare the gene expression between patients without schizophrenia and those with schizophrenia, see which gene expressions are differential in the sense whether they're upregulated or downregulated with when compared, which may serve as a marker to predict when someone has schizophrenia or if they're going to have it, or it may also help in a way to regulate treatment. So one of the main areas that's damaged with schizophrenia is the amygdala. So the amygdala is a portion of gray matter located at the anterior medial area of the temporal lobe that is involved with emotional and behavioral functions. The basolateral portion of the amygdala may play an essential role in normal behavioral response to environmental stimuli. So amygdala disruptions may cause emotional disturbances in schizophrenia patients, have a direct link to memory function damage, and have a direct link to decrease of emotional processing and various disconnection from individuals.
Cyverse is a computing resource funded by NSF and hosted by University of Arizona that allows university students to conduct biostatistic projects. We used Cyverse to run an RMTA analysis to cross-examine the genomes found in the amygdala of schizophrenia patients to the human genome and chromosome 1. It was used to analyze the statistical difference in read counts of differentially expressed genes, or DEGs, to determine if they were up or down regulated.
Unfortunately, Cyverse was not able to analyze our data. We used data from the NCBI. It was from the amygdala of schizophrenia patients, and the file sizes were pretty large. They were around somewhere between 4 gigabytes and 7 gigabytes. And we think Cyverse may have had issues computing that large of a file. And we were using multiple files like those, and we had to compare it with the human genome and chromosome 1. So even the human genome was close to 1 gigabyte. And we think Cyverse just did not have the computing power to compute it all in a timely fashion. And at one point, a week worth of analysis was canceled when they went under maintenance, and they canceled the entire analysis. So what we did expect to happen when compared with the human genome, there would have been an upregulation of genes associated with immune pathways and function in schizophrenic patients. And there would have been a significant expression differences in the amygdala regions between cases and controls. So even though we did not get the DEGs we wanted to determine which differential expressions occurs with schizophrenia patients compared to controls, we still wanted to figure out what may have caused such differential expressions. So one of the key areas that we looked at is the disrupted in schizophrenia 1 gene. There is a Scottish family, a 21-member extended family, and their data is online, a karyotyping data, which shows a translocation in the chromosome. It's on chromosome 1, between chromosome 1 to 11. And as you can see, that's the location area, that 1 to 11. And the Q42.1 is usually it's referencing the long end of the chromosome. And it occurs between exons 8 and 9 breakpoints. This was figured out using karyotyping data, comparing the family members that didn't have it and the ones that did. Usually when there's a balanced chromosomal translocation in that area. Usually there's bipolar disorder, schizophrenia, and other various mental disorders that occur, such as depression. So at that breakpoint, at the translocation breakpoint, it is usually associated with the encoding of abnormal protein, resulting in mitochondrial dysfunction. And the left temporal lobe had reduced cortical thickness in various MRI scans of that area with people who have the translocation. There's usually decreased glutamate levels in the prefrontal cortex and increased activation of the caudate nucleus. So ultimately, the determination of genes differentially expressed for subjects with mental disorders such as schizophrenia can be used to predict if someone is predisposed, such as the use of blood analysis. So it may be good to figure out a way to accurately detect if someone is predisposed to it. And because schizophrenia is irreversible, you just don't want to trigger it. And some people may be able to plan ahead and learn coping skills for stressful events, because it's usually after they suffer a stressful event or a long-term stressful event that tends to trigger schizophrenia. And considering the importance of the mitochondria, such as its use as energy for cells, immune functioning, maintaining homeostasis and regulation of apoptosis, when that translocation occurs, it does affect mitochondrial function. And mitochondrial function affects immune function. So when someone goes through a stressful event, the brain treats it almost as an injury. It releases hormones such as ATCH and cortisol. And those hormones, they trigger immune responses. And when the mitochondria isn't functioning properly due to dysfunction, it may affect how the immune response is, which results in attacking good cells. And it may explain why there's 25% less gray matter or white matter in patients with schizophrenia.